Later.
Yeah, I think it started.
Okay. So I would like to welcome everyone, and it's my great pleasure to introduce Dr. Eryn McFarlane. Eryn and I did our PhDs together in Anna Kvarnström's lab in Uppsala in Sweden, and Eryn has since gone on to work at the University of Edinburgh with a Marie Curie fellowship on deer hybridization, and she's now working on this and some other projects with the University of Wyoming. So with that, I would like to say thank you very much, Eryn, and take it away.
Thanks so much, Will. Thank you for having me. I should say it wasn't a Marie Curie, it was a Swedish Research Council grant, which was great because it was more time.
See, of course, I forgot that. Thank you.
No, that's okay. Thanks so much for having me. I am excited to tell you a little bit about my work today on anthropogenic hybridization between red deer and introduced Sika. As Will said, I'm now working at the University of Wyoming, but I am physically in Canada, which has been great to have a fully remote postdoc. Whoops. So I work in secondary contact, and secondary contact, when two species come together, can be used to study the speciation process. And it's a fantastic natural laboratory because when we have these species that are evolving in allopatry and we don't know, unless they're tested together, we don't know anything about how far the reproductive isolation has gone. And so there's a number of possible outcomes at secondary contact. There's competitive exclusion, there's character displacement, and there's hybridization. And for the last few years, I've been studying hybridization and the resulting recombinant individuals to understand a little bit about the possible causes of this breakdown of reproductive isolation and then the genetic consequences of it. And one of the reasons that I do this and that I've been supported in doing this is that secondary contact with hybridization is expected to be a lot more common with human disturbance and with climate change, for example. And so, in general, I study the ecology and genetics of speciation, focusing on secondary contact. And most of my work recently has been on anthropogenic hybridization. So this is hybridization that's happened in the recent past that's due to human impact. So whether this is urbanization or climate change or the translocation of one species into another, this is what I'm working on right now. And it tends to be very recent, usually, but not always, since kind of the Industrial Revolution. And the reason that we're interested in doing this is because the introduction of non-native species can lead to a decline in native species. And whether that is due to any of the three things that I said, competitive exclusion or hybridization, but once they start to hybridize with each other, we can have individuals in the population who look like the pure species and who fill the niche of the pure parental type, but that are carrying the alleles of the introduced species. And this is a way that we can lose biodiversity because we can get this allelic replacement. And this is a form of human-mediated evolution because the allele frequencies are changing in our native population. And one thing that I always like to say really loudly at the very beginning of my talks is that official policy often doesn't cover hybrid individuals. This is the case in the United States. This is more or less the case in Canada and Europe, where hybrid populations are taken on a case-by-case basis, where hybridization might be considered when looking at some kind of species at risk assessment, but it's determined on a case-by-case basis whether or not the hybrids are having a negative impact. And in many cases, if there are hybrids in the population, it's just not going to be protected at all. So I always have these canids at the bottom of my talks where I have on the left-hand side coyotes, on the right-hand side we have gray wolves, in the middle we have these red wolves. And red wolves were one of the first species that was protected in the American EPA. They show up in the American Southeast. And so there's really large swaths of land that are protected to protect these red wolves. And we know that they hybridize with coyotes, and we know that through red wolves, coyotes can hybridize with gray wolves. And it's only using genomic data that we've seen the potential that actually the red wolves are an example of a hybrid species. But if they're still hybridizing with both species, then they're not a very good, with both parental types, they're not a very good species. And so there's been quite a lot of lobbying in the States to have them assessed as not their own species, and then they would no longer be protected by the Endangered Species Act, which again means that we have this biology kind of intersecting with policy, and then it's really intersecting with our power to detect hybrids. And before genomics, we didn't really know this in the same way. And that's a very simplified version of that story. So I talk a lot about backcrosses and I talk a lot about hybrids. And so we can define a backcrossed individual. And so I have here a pedigree of a little brown spotted deer and a big red deer who are breeding with each other. Their first offspring is an F1 individual. And then when that F1 breeds back into the red deer parental population, they're making a first generation backcross. And so there's a couple of cool things to note. The first is that while an F1 is going to be really 50-50 of the fixed differences between the little brown deer and the big red deer, the first generation backcross is on average 75% big red deer, 25% little brown deer, but there's a distribution around that. So some individuals are going to be much more red deerish like, and that is the case all the way down the pedigree. That while on average the next individual, the second generation backcross, it's going to be 87.5% red deer, there's a distribution around that. So the other thing to keep in mind is that all offspring of an admixed individual are themselves hybrids. And so if all things are considered equal, if there's no selection against hybrids and no sort of mating, then the number of hybrids in a population will always increase, because anytime a hybrid breeds with a pure species individual, you get another hybrid. So there's some questions that we can address in anthropogenic hybrid zones, and these are some of the things that I'm going to address today. The first question is, how many animals in the population are hybrids, and how can we detect them? And spoiler, we use genomics. The second question is, are there loci under selection or that are associated with these diverged phenotypes? And then I'm going to spend a little bit of time talking about what the next steps in anthropogenic hybrid zones are, or what I think they are. Okay, so how do high density markers change the power to detect hybrids? So these backcrossed hybrid individuals must be differentiated from both the parental types, our pure species parentals, and those individuals who are at the 50-50, the F1. And because of this, we need many more markers to detect them than we would to just detect individuals that are exactly halfway between the parentals. But unfortunately, up until 2016, I guess it's getting to be a little bit of a dated reference, 20 markers was average for study of anthropogenic hybridization. And as a result, our individuals who are backcrossed past that first generation of hybridizing were often improperly assigned, and they're easily lumped in with the parental species. This is a problem because if we have our hybrid individuals that are lumped in with the parental species, we might not cull them if we go out to cull hybrids, and we might inadvertently bring them into a breeding program if we're setting up a captive breeding program. And both of those cases would mean that the hybridization would continue. So what we did was we extended a deterministic model that was put out by Boecklen and Howard to ask how often will a backcross be undetectable from a parental species. And this is banking on the understanding that there is a distribution around that 75%. So if you'll forgive me, if you'll bear with me, I'm going to bring you back to your Punnett square days, where we have our parental gamete alleles along kind of the y-axis there, and then an F1 gamete alleles on the x-axis. So this is, in the top plot, we have a one locus system where we're only typing one locus, and they're fixed at little a for the parental types, and either big A or little a is possible for the F1. And then again, below that, there's the two loci system, where again, parental types are fixed at little a, little b, but the F1 gametes can have either big A, big B, big A, little b, little a, big B, or little a, little b. And the point of what you take from this is that either in the one locus system, the little a, given on by the F1 gamete, or the little a, little b, that is indistinguishable from the parental type. And so what you can see here is that when we've got our first generation backcross, in a one locus system, we have one individual who we genotype at this locus is completely indistinguishable from the parental type. That's the little a, little a. So half of our individuals are indistinguishable from the parental types at this locus. And if we have a two loci system, then 1/4 of our individuals are indistinguishable, which makes the point that the more loci that we have, the more hybrids we're going to be able to detect, or the fewer hybrids are going to slip by our detection. So we extended this model to kind of the genomics era, and we extended it for 10, 100, and 1,000 diagnostic markers, so markers that are perfectly fixed between the peer species, over 10 generations of backcrossing. And so what I have on this plot here is each of the lines is 10, 100, and 1,000 diagnostic markers. Backcross generation is across the x-axis that goes from 1 to 10. And then the y-axis is the proportion of genetically undetectable hybrid individuals. So this was 1/2 in the 1 locus system and 1/4 in the 2 locus system for a first generation backcross. And then the red line is denotes 80% of individuals. So this is when we get beyond 20% of individuals that are undetectable. So this is our good power line. And what I want you to take from this is that very quickly, as we get more generations of backcrossing, we need more diagnostic markers to be able to detect our backcrossed individuals. So after 3 generations of backcrossing, which is what this pedigree looks like, we have good power with 10 diagnostic markers. We have good power with 100 diagnostic markers after up till about 6 generations of backcrossing. And we have good power to detect up to 9 generations of backcrossing with 1,000 diagnostic markers. And I was teaching this in a class yesterday, and I got asked, yeah, but how often do you see 1,000 diagnostic markers? And the answer is very rarely, even with high density sequencing. So it's not common. So what this means is that most studies of anthropogenic hybridization are systematically underestimating the proportion of backcrossing. If you don't have many, like hundreds of diagnostic markers, then you're not really able to see past 5-6 generations of backcrossing. And this is what's been found empirically in wolves and trout when folks have gone back and retyped the same individuals that they'd already assessed using something like microsatellites. If they then go and type them on high density genomic markers, they find that they are reassessing a lot of individuals as hybrids that before they were calling pure species. Okay, so from there, I promise I'm going to talk about deer. We can ask how many hybrid deer are there in Scotland? And so I work in an anthropogenic hybrid zone between red deer and Sika. So in this photo here, you can see we have a male Sika in the foreground and a male red deer in the background. And the Sika were introduced first to Ireland and then to Scotland. And they hybridize in Scotland. They hybridize in many places across Europe, which I'm going to talk about a little bit later. And the red deer especially are an economically and culturally important species in the UK. So they are hunted. They're on the like big 5 of species to see if you're ever in Scotland. They're very beautiful, and they're very much a part of the culture. And so this system has been well studied since the 1990s. By Josephine Pemberton, who's my postdoc host, Helen Sen, and Steph Smith before me. Oops. So to just give you a little bit of a timeline, I can't tell if my little Zoom window is covering people's face. Anyways, this is the Viscount Powerscourt. He's the person who brought the Sika over from Japan to Ireland originally. So he was a member of the aristocracy, and he thought that the Japanese Sika were very pretty. So he brought them over to Ireland in 1860, where they promptly started escaping. They were introduced from Ireland to Kintyre, which is a part of Scotland I'll be talking about, in 1893, and there were known escapes during the First World War. The first putative hybrids were seen in Kintyre in 1970, but again, these were phenotypic hybrids. And so what I've listed below here on the timeline at the bottom are a number of papers that have been written on this subject, using genetic markers, starting with allozymes. So starting with 2 allozymes and 2 microsats, and then moving up to 11 diagnostic microsats, 22 diagnostic microsats, and then finally 45,000 SNPs. And then increasing the number of deer. And so we've been sampling these deer since the 1990s, and then in many cases resampling deer as we've gotten more genetic methods. So when I'm talking about Kintyre, I'm talking about a region of Scotland that's just southwest of Glasgow. So you can see I've got here marked Campbelltown, and we have sampling sites all up and down this Kintyre Peninsula. Yeah. And as I said, we had 45,000 markers, so we did use a snip chip approach, and we estimated admixture estimates. So this is the proportion of red deer from 0 to 1, or a Q score, using admixture. If you've ever used structure, it's the same algorithm of structure, and it's consistent with point estimates from past structure. What's great about admixture and why I prefer it is that it is very fast, and it gives species, we can get credible intervals and do our species assessments using those CIs, which means that instead of looking at the point estimates and having to use a threshold, we can look at whether or not our confidence overlaps 0 or 1. And so if an individual has a credible interval that overlaps 0, we say that it's a Sika. If it has a credible interval that overlaps 1, we say that it's a red deer. And if it has credible intervals that exclude both 0 and 1, then we can say that it's a hybrid. So here is what we found. The x-axis here are the individual deer. So there's 513 deer on this plot. The y-axis is proportion red deer, so from 0 to 1. And individuals in blue are those individuals that we assess to be Sika. Individuals in red are those individuals that we assess to be parental type, pure species red deer. And anybody in purple is somebody that we assess to be a hybrid deer. And this is just another way of looking at the same distribution, where you can see that really most of our animals are very red deer-like or very Sika-like. So what is on this plot here is the number of individuals on the y-axis, and then the proportion red deer, the Q score, is on the x-axis. And so you can see that we really do have a U-shaped distribution, where most individuals are very red deer-like or very Sika-like, and then we have hybrids kind of all across the distribution there. So what I'm going to show you to really just make the point about the differences between using these genomic markers versus using the 22 diagnostic microsatellites. I'm going to show two plots. The top plot are individuals that were assessed using 22 microsatellites to be red deer, and the bottom plot are individuals who were originally assessed to be Sika. And so, as I said, all of these individuals were thought to be red deer, and you can see that there's a number of purple individuals in there now that with higher density markers and with more confidence we can assess to be Sika, or to be hybrids, sorry, it's early. And then the same was true of individuals who we thought were Sika. We can then see that in fact a number of them were hybrids, and we just didn't have the ability to detect that before. Yeah, so this is, we had originally 113, I think, 113 hybrid individuals of the 513. And we ended up assessing using genomic markers 222 hybrid individuals of that same 513. So the average hybrid score overall was 0.614. So what this is saying is that we really have hybrids across the entire spectrum from 0 to 1. And so this did span from 0.008 to 0.992. So we have hybrids who look very much like Sika and have a lot of most of their genome is Sika, and we have hybrids who most of their genome is red deer, but we can still detect that they are back-crossed. And this is consistent with 6 to 7 generations of back-crossing when we take into account the generation time, which lands us right in the 1970s, which is pretty great given that that's when the first phenotypic hybrid was seen. We don't find evidence of any F1 individuals, so while we do have some individuals whose Q score is near 0.5, those individuals were not uniformly heterozygous at our diagnostic markers, so we ruled them out as F1s. Another kind of interesting point is that we had 9 individuals that had credible intervals that overlap 0, so we called them Sika deer, but with red deer mitochondrial DNA. So these were animals who obviously had hybridization in their ancestry, and it must have been past that 6 to 7 generations of backcrossing. So we can see that they'd had a red deer mother at some point, red deer having mitochondrial DNA at some point. But if we'd used just our nuclear genome, we wouldn't have been able to see it. So that's kind of a cool point, that like even with 45,000 markers, there's still more than we can detect there, more hybrids than we can detect because of how back in their history it's going. Okay, so we can then ask, are there loci under selection or that are associated with diverged phenotypes? And one of the most diverged phenotypes between the two species is Carcass Mass. There's really a large size difference between them. And I don't think that I mentioned it before, but all of the deer that I sample are called deer. So this is a population that is sampled by the Forestry Commission of Scotland. They take a carcass mass, they sell the deer for meat, and I get a sample for genomics. So they are unbiasedly sampled, but it means that we're not, this is really the only phenotype that we can get for them. And so carcass mass is very highly diverged between species. So what I have here on the x-axis is carcass mass in kilograms. This is about 2/3 of live mass, just for those who are interested. And then this is a density plot. So again, I have Sika in blue and red deer in red and hybrids in purple. And you can see that there's two distinct peaks for the Sika and the red deer. Red deer can be easily twice the size of a Sika. And then the hybrids have a distribution that kind of covers both. And we also have many red deer that are kind of way out here in the tail that can get very, very big. To look at this another way, so this is along the x-axis, this is our proportion red deer again, and then carcass mass along the y-axis. And you can see that our Sika, who are at 0 proportion red deer, are really quite small. Our red deer can be really quite big, quite a lot bigger. And then hybrids run completely the gamut between both. And so we can take advantage of these recombinant individuals to start to ask something about the genetics of carcass mass. Oh, I just wanted to show this picture of a red deer male in the background and then a Sika male in the foreground to again show the kind of massive size difference. And then, so I am actually going to show a picture of a culled deer. So if you're squeamish, I will tell you when it's gone. It's not too graphic, but just I want to give you an opportunity to kind of close your eyes. But this is a picture from one of my hunters. And so this deer here was shot in our Kintyre region. That's a Labrador on it. That's probably a 20-kilo dog. This is, I'm trying to remember, this is like a 90-kilo deer for carcass mass. But what I want to point out is that it's actually not that red, and it's got tons of spots right on its butt. So this is very likely to be a hybrid deer, and especially where it was caught, it's very likely to be a hybrid deer. And it's huge. It's right up here in our distribution. Okay, the deer is gone. So I used admixture mapping to look for QTLs, so quantitative trait locus, that are explaining some of the variation in carcass mass. And using admixture mapping, we can talk about the number, distribution, and identity of genetic markers that explain variation in a quantitative trait. And what's great about QTL mapping, it makes it very powerful compared to other types of genome-wide association studies, is because it makes use of the recombinant genomes of hybrid individuals. And so if you're thinking about kind of genetics on lab animals or fruit flies, admixture mapping is like our F2 crosses. We can take individuals that have a very similar overall admixture score, but we know that there's lots of phenotypic differences between those individuals, and that's indicative of that even if they have the same overall admixture score, different parts of the genome they have ancestry at different parts of the genome from the two species. And so it makes a great comparison because we can look for those long tracks of the genome that can be associated with mass in this case. And additionally, it's like in the deer and in general, because we have this huge, like extreme phenotypic differences between them, it means that there's an opportunity to really kind of levy those differences to look for genomic markers. And indeed we found some. So you can think of this as kind of like a Manhattan plot. So we have the service linkage groups on the bottom here from chromosome 1 to chromosome 34. Chromosome 0 are things that were unmarked in a linkage group. And then the y-axis is effect size in kilograms per allele, which means that for an individual like, say, this on chromosome 9, where it's about 0.75 kilograms per allele, that means an individual who is a heterozygote is on average 0.75 kilograms heavier, and homozygote is 1.5 kilos heavier than a homozygote in the other direction. And we did find, so here is our threshold of acceptance, where we are saying anyone above this threshold, or any of the markers above this threshold, we accept that these are markers of large effect. Because I did this in a Bayesian framework, we have what's called the proportion inclusion probability. But again, you can read this kind of like a Manhattan plot, where we have linkage groups along the y-axis, and then the proportion inclusion probability linkage groups along the x-axis, and then proportion inclusion probability along the y-axis. And anything that is included in the sparse matrix, so anything that we think is a marker of large effect more than 10% of the time, we then include in our analysis. And you can see that there's a number here. We ended up having 10 markers that were included, that 10 markers that 10% of the time in the Bayesian analysis we thought were markers of large effect, and that had a more than 10% proportion inclusion probability. And these spanned across the genome and were associated with 45 genes. And then when we looked at those genes, I'll come back to that. And then when we looked at those genes, we found that of the 45 genes, I think it was 13 were associated with either mass or BMI in humans, or height in humans. Okay, I'm going to show a figure, and I'm going to explain it, and then I'm going to show it in context and in a panel. So what we did for each of those SNPs was we asked whether the SNP that we had found in our overall analysis that I just showed you, whether the SNPs that we found that were associated with carcass mass were associated in the direction that we expected. So the previous analysis just asked whether there was variation, it's a regression essentially. So just ask whether or not there's variation in mass that's associated with variation in the SNP, in the genotype. We wanted to know whether or not that variation was polarized, where each of those SNPs, if you had the smaller genotype, were you more Sika-like, or was that genotype more common in Sika than if you had the larger genotype, which is what we would predict, but is not necessarily what would have happened. And so what we have here is, for one SNP, I have females for one SNP where we have the genotype of 0, 1, and 2 copies of the allele, I then have mass along the y-axis, and I've split it into females and males because there is a very large size difference between females and males. And then we look for each of the individuals that have each of these genotypes of 0, 1, 2 copies of the heavy allele. We then looked at what the mass was. And what we've done is we've always polarized so that 2 copies of the allele is 2 copies of the red deer-like allele. Not sure I explained that as well as I wanted to. What I can tell you is that essentially all of the SNPs follow the same pattern, where the light allele was at higher frequency in Sika than the heavy allele, and the heavy allele was at higher frequency in red deer than the light allele. And so you can see for all of these plots that, with the exception of the one that's on the X chromosome, which I'm not going to talk about at all, that they all kind of go in the same direction, suggesting that in addition to being associated with carcass mass, these are all markers that are somewhat associated with ancestry. And so we found markers that are not just associated with being big, but being associated with being big in red deer, if that makes sense. Okay. So the next question that we asked is, when we're looking at locus-specific parameters are the genomic regions that are introgressing at extreme rates. And the reason that we wanted to ask this, that I think I kind of mentioned before when I was talking about anthropogenic hybridization generally, is that while we can get pretty concerned about any population where there's only hybrids in the population, if you have only hybrids in the population, but the allele frequencies are the same as the two pure parental types, then while it is still concerning, you haven't really lost any genetic diversity because all of your alleles are still there. And so there's some kind of high profile cases of breeding hybrids together to try and reconstruct parental types that no longer exist. And they've done this or they're starting to do this very slowly, as one can imagine, in Galapagos tortoises, where they had an extinct species, but they have a number of hybrids, so they think they can kind of get it back. It's pretty cool. And so one of the things that we found is that there's variation in FST across the genome. So the differences between the parental types are not the same across the genome. There's heaps of variation. And this just shows FST along the first 10 chromosomes. And I hope that you can see that this FST varies a lot across the first 10 chromosomes. And so we wanted to ask if there were alleles at specific, we wanted to ask if there were loci that had alleles that were introgressing faster from one species into the other or back than we would predict based on kind of the genome-wide rates of introgression. And so this is a plot that I've taken from Gompart and Berkeley, where we can look at locus-specific introgression using what's called a Bayesian genomic cline. And so what's along the x-axis here is the hybrid score of a hypothetical individual. So this is the same thing we've been looking at the whole time, where 0 is a Sika and 1 is a red deer. And then we can ask for each of those hybrid scores of a hypothetical individual, what is the expected probability of ancestry at a specific locus? And so if you have introgression at the exact speed that you would expect that is consistent with the whole genome, then at a 50-50, so at an F1, you have a probability of ancestry of 0.5. If there is, in theory, if there is selection against heterozygotes, so kind of selection against hybrids at this locus, then you would expect a very, very narrow cline where you have very few heterozygotes, even if you have a hybrid score of like 0.2 or 0.3. Whereas if there is no selection at that locus against hybrids, then you can expect a little bit of a wider cline where the hybrid score is consistent with the probability of ancestry. And then if there's selection for heterozygotes or conceivably adaptive integration, then you would expect a wider cline where even if you have quite back-crossed individuals, you're still predicting a heterozygote at that specific allele. So yes, so we can start to delve into selection at specific loci for hybridization based on these genomic clines. And so we did this in our red deer, and we found heaps of differences among the loci. So we found a number of loci with positive beta. So this is this very steep slope that it kind of seems like evidence against introgression here. So we found lots like that. We found many markers with a negative beta. So here is this very, very wide slope. I should say that the differences in color here are whether there is whether the introgression tends to be from red deer to Sika or Sika to red deer. So that's another thing that can vary is the intercept. But mostly I talk about slope. So we have the very steep positive beta, which suggests that there's selection against introgression at that locus. We have the very wide negative beta, which suggests if anything there's selection for introgression at that locus. And then these are some of the markers that we're most interested in. There's about 60 of them, where there is a negative beta and a negative alpha. So here we're getting introgression faster than we would have expected based on the genome-wide average, where alleles are moving from Sika into red deer. And so if we were going to have conservation concerns about this system, these are the regions that we're going to see allelic replacement first, because here are regions that we are having alleles move very quickly, faster than the genome-wide average, from Sika into our native species of red deer. Okay, so what have we learned about deer hybridization and introgression so far? In Kintyre, in the region of Scotland that I mostly study, we've detected 26% more hybrids than we knew about before. And this demonstrates the need to include more markers in this and likely many other systems. We found 10 markers for Carcass Mass. I should say those 10 markers were not among those that were introgressing at very fast rates. So they were introgressing at rates consistent with the genome-wide average. Of the 45,000 markers, 60 are extremely concerning. These are the ones I just mentioned. So these are instances of Sika allele that could soon replace red deer alleles in this region. And these markers should be considered if we choose to examine gene-based conservation in Scottish deer, which we won't. Red deer are really a species of least concern, and they're all over Europe, and they're fine. So what are our next steps in anthropogenic hybrid zones? I hope you'll bear with me if I have time. Yeah, I can do it. While I take a little detour into simulation land, because there's some questions we want to ask about anthropogenic hybrid zones kind of in a more general scale. So when we were looking, I oversimplified quite a bit when I say that introgression, fast introgression is associated with selection for hybrids at that locus, and slow introgression is associated with selection against. That would be great if it were that simple, but of course these are natural systems, and so there's drift. There's lots of genetic drift, and when we were doing these locus-specific cline analyses, we didn't really have a good idea of what the null expectation was for how many markers are going to be introgressing really quickly and how many markers were going to be introgressing really slowly without kind of invoking selection. And so I did a number of simulations in SLIM where I varied the number, so I was simulating genomes, where I varied the number of generations since admixture began. I varied the rates of migration and interbreeding between the populations, and we have population ratios of 1 to 1 or 1 to 3. What is important, if nothing else, when I show these graphs, is that I did not simulate selection at all. So there is no selection either for or against hybridization at any of these loci that I'm going to show you. And so here's the kind of graph that I'm about to show you a ton of. So this is representative of one of the simulations that's been done many times. I'm sorry for the loci category. It's a little bit confusing, but what I want you to know is that the very middle green point, that is not significant, not significant. So anything other than this this green point right in the middle is evidence of either positive or negative introgression or a change in the intercept of the cline. Okay, so anything other than this one right in the middle could be taken as evidence of selection, but again, I haven't simulated selection, so there is none. Okay, this is a monster plot. On the x-axis is increasing amount of admixture at secondary contact. On the y-axis is increasing from top to bottom, is increasing number of generations since secondary contact. So the top row is 10 generations, the middle row is 100 generations, and the bottom row is 1,000 generations. Across the rows, we have slow, medium, and high rates of hybridization. So that's the letter in the middle. So the first two columns are slow migration, hybridization. The middle two columns are medium rates of that interbreeding, and the last two columns are high rates of hybridization. And then it just alternates for whether or not it was a 1-to-1 or 1-to-3 ratio. This is a lot. What I want you to take from this is that in almost all cases, there are lots of markers that are significantly diverged from the case of not significant, that they have variable alphas or variable betas that are different from the genome-wide expectation of not significant. And what this means is that we can get lots of these, these are regions of the genome that really are introgressing quickly or faster than we would expect based on the genome-wide expectation. But it's not because of selection, it's just because of drift. It's just because we have an expectation of this scatter shot. And so this is happening in all of our scenarios, that there are regions of the genome that are introgressing faster than you would expect based on the genome-wide expectation, which means that without accounting for drift, we really should be very careful claiming selection either in the positive or negative just because of rates of introgression. So the next thing that we were hoping that we could do is we were hoping that if we had replicated hybrid zones, if we looked at hybrid zones all across Europe or what have you, we kind of said, okay, drift happens, that's fine. Let's hope that drift is idiosyncratic and that selection would be consistent across populations. And so if we're getting very fast introgression in one place, that could be due to drift. But if we're getting very fast introgression across the board, probably that's selection. We kind of made this verbal model. We're not the first people to make it. So we decided to simulate this, and we decided to ask how often replicated hybrid zones resembled each other. So we simulated, here it's a stepwise model, so you can think of on the red side, 100% red deer, on the blue side, 100% Sika, that they then stepwise together to meet across these demes, and then at zeme 6, they're hybridizing with each other. And so there's 150 diploid genomes in each of these populations. And we replicated this 20 times, and we varied migration rate, selection, and the type of genetic incompatibility that was leading to selection against hybrids. And I'm sure that I'm completely out of time, so I'm going to try not to go through this quickly because I think that that's a recipe for disaster, but I am going to try and get through it. Right. And so again, I'm going to show one plot, and then I'm going to show lots of plots. So what we have here on the x-axis from 0 to 1 is, again, proportion. You can think of it as proportion red deer, where 0 is a Sika and 1 is a pure species red deer. And then these are density plots. So 12 is kind of the number of individuals. Each of these lines is an independent replicate of the same simulation. So each density plot is one of our 20 populations. Here we have very low migration and no selection. Okay, and I think you can see even in this case where there's very low migration and no selection, there are differences between the populations. I can tell you that they are statistically significant, but biologically they probably don't matter that much. Okay, if we look at the entire simulation, again, we have admixture proportion along the y-axis and density along, admixture proportion along the x-axis and density along the y-axis. And then the first 3 columns are migration rate, a low migration rate of 0.01, and then the second 3 columns are a faster migration rate. And then we have C is our selection, so we have C equals 0, C equals 0.2, and C equals 0.9 for 1, 2, 3, and then 1, 2, 3. And then we have different types of genetic incompatibilities, either Dubinsky-Muller incompatibilities, those that interact with the environment, what's called pathway incompatibilities, and then pathways that interact with the environment. And what I want you to see here is first that there's lots of variation among the different simulations and how much differences there are between populations. So if you look, for example, up at this case here, where we have slow migration but lots of selection against hybrids, you can see that there are heaps of differences among populations. And so these are cases where we've simulated the exact same thing and we're getting hugely different outcomes. And so those different outcomes are due to drift. They're not due to differences in selection. They're not due to differences in the genetic incompatibilities or anything like that. They're just due to differences in sampling error. And so this is something that we need to keep in mind when we are setting up our studies of replicate hybrid zones, for example, that we're going to have to account for the differences between populations. So we both need to account for the fact that we can get heaps of variation in introgression due just to drift, and we can get heaps of differences among populations just due to drift. So hybrid zones in general are rife with sampling error and drift, because in general that formation of the F1 is just not going to be that common. And then one other point that I can make is that we see huge differences kind of across the genome. So it's not just at points where we have where we've simulated the incompatibility. So this is, again, our DMIs and our pathways. I haven't shown the environmental differences here because it doesn't really matter. And then looking at all of the populations are, again, in the same colors as they were before, looking at chromosomes 1, 2, and 3. We did simulate 10 chromosomes, but these kind of show the pattern. And you can see, again, that when we have low migration and strong selection, that you get huge differences in ancestry across the genome, that you're just getting this massive variation among populations that should be the exact same, or that we expected to be the exact same. Okay, so all that to say is that we were really hopeful that replicate hybrid zones could be used to differentiate between selection and drift, but we are now seeing that these extreme rates of selection could be the results of selection or drift. And we were hoping that if it was not replicable, then it was likely due to drift. But based on our simulations, that we're just not at all sure about that anymore. We are hoping to include the modeling of the simulations and what we know into analyses of replicate hybrid zones to be able to account for those among population differences, while still looking at rates of introgression to try and kind of pull apart selection and drift. And to do this, we have sampled 3 regions in Ireland, 20 in Britain, and 11 so far in France. And I am communicating with folks in, say, the Czech Republic to have more samples. So empirically, I'm still hopeful to be able to understand how selection and drift are interplaying across the genome when we have anthropogenic hybridization. But it is going to be a little bit harder than we originally thought it would. So in conclusion, we need more markers and studies of anthropogenic hybridization. I believe that the next steps include replicate hybrid zones, but it's not going to be as simple as I kind of naively thought. And just generally, I always love to throw this in, that we need to consider biology rather than statistics when we make policy decisions, because even as we have increased detection of things, the question that I always get is, does it matter? Does it matter if we can detect 10% back crosses? And this is where we come back to this like case-by-case policy making, where it really depends on the system, and it depends on the natural history. It depends on what's important to the kind of stakeholders in the region. And so once we have high power statistics to be able to detect kind of whatever we want, then we can take a step back and consider the biology. And maybe we say, like, actually, we're thrilled with hybrids running around because we're thrilled to have anybody running around. And those are biology and policy decisions, not statistical ones. And I hope that I've convinced you today that instances of secondary contact are excellent natural laboratories to understand the speciation process, and that we can go from asking questions about deer and the size of deer and whether or not individuals are hybrids, to kind of more mechanistic questions about rates of introgression, all in these same empirical systems. And that we can then address some conservation concerns while asking kind of blue sky science questions about speciation, and that by using genomics we can do this in a non-model system. And this work has hardly been done in a vacuum, so there's lots of people that I need to thank, including various funding sources over the years, and then lots of people who have been super supportive of my research and my want and need to chase deer and their genomes. So and thank you for listening, and I'm sorry if I went way over time.
Eryn, thank you very much for that. That was absolutely fascinating. If anyone has any questions, then feel free to unmute yourself and ask them. In the meantime, I have a question. Can you hear me okay? Great. So those 60 markers that were doing crazy things, do you know what they're associated with, phenotypically or anything like that?
Okay, so I made a very tactical decision not to look. And the reason that I did this was because I am not convinced that they are the result of selection at all. What I can say is that they're not the markers that were associated with Carcass Mass. So they're not those, because we do know those. But I did not want to go and look at any of the kind of human or cow data that I could have pulled to say that this is associated with this or that or the other thing, because I was afraid that I would be very tempted into storytelling if I did that. And so the party line as of right now is that those markers are probably introgressing that quickly because of drift. Which is fine. Like, they are still introgressing very quickly. We still could get allelic replacement at these genes of Sika of Sika alleles, and that can be concerning even if we don't know what they do. But I was very afraid that I would go in and find something kind of vaguely interesting about, these genes are associated with, I don't know, living with humans or whatever, and I would make a whole story about it. So I've never looked. Which okay. I get asked this almost every time. So maybe I should look.
So I've never really thought about it before, but I guess it makes sense then that drift can sort of act at different speeds. So that could easily just be that then. Okay.
Yeah, that's really it, is we know that just because of sampling error, and because especially in a hybrid zone where not all animals are hybridizing, it really is a finite number of individuals who are involved in this, and it might be like it's probably thousands in Kintyre, but it's still finite, which means that kind of sampling can result in regions of the genome that are introgressing faster just by who's breeding. And if we don't account for that, then I at least would be really tempted into storytelling about selection. And we're seeing there's a little bit of a pullback right now in the literature about maybe not invoking adaptive introgression without additional evidence for it.
Okay, thank you very much. Are there any other questions? If not, I have one more. I'm not going to let you escape yet. What was I going to ask? Oh, yes. So how have any of your results filtered down into policy making yet, or do you expect them to filter into any sort of management happening in the United Kingdom?
It's a really good question. So we have talked to people in the deer management community about the hybrids. They are uniformly unconcerned. There are so many red deer in Scotland that people aren't really worried. We can shout it from the rooftops that this hybridization is insidious and it's going to be impossible to stop. And we can see that, especially in Kintyre, where we have very high rates of hybridization, we can almost see it moving into the mainland, like out of the peninsula. So there's more and more hybrids. Most of the deer management right now in Scotland, at least, is just managing for forestry. So they just call to try and keep the densities down to a certain amount. My postdoc host Josephine has literally suggested, fencing off Kintyre to try and stop hybrid individuals from escaping. But I don't think nobody so far. very few people but us care. And so I fully expect in 200 or 300 years to have a very great I told you so moment when all the deer in Scotland are hybrids, but nobody cares yet. So, yeah.
Okay, thank you very much. Any other questions? Okay, with that, I would like to say thank you very much to you again, Eryn, for joining us, and especially for joining us at such an unsociable hour for you. I think it's just turning 7 in the morning for you now, I think, isn't it? So, yeah.
Thank you so much for having me.
Yeah, thank you very much. And yeah, speak to you soon. All right, thanks everyone. Bye.